Hello and welcome to our introductory webinar about gene therapy, what it is, its promises, and challenges for hypophosphatasia. This webinar is made possible in part by Aruvant Sciences, and we're grateful for their support. I'm Deborah Fowler, president of SoftBones. We're thrilled to have you from attendees from around the world joining us on today's webinar. Before we get started, we will do a few Zoom housekeeping notes. If you have any technical issues during the call, please enter them into the chat or email us at info@softbones.org. Any questions should be entered into the chat. We will hold a Q&A session at the end of both presentations. This webinar is being recorded so you can come back, listen again, invite others to join in and listen at a later time. It will be shared on our social media channels and in our SoftBones resource library, as well as on our YouTube channel. As you may know, research is core to the SoftBones mission. We hear a lot about gene therapies in the news and in social media, and now they're being used in diseases like HPP that are caused by genetic mutations. We are excited to share the progress that's being made in gene therapy and to lay the foundation of understanding of what this means in hypophosphatasia. I'm excited to introduce you to our speakers today. Dr. Matthew Grahl is an assistant professor and Tier 2 Canada Research Chair in Musculoskeletal Biology and Health at Western University in London, Ontario, Canada. His research program investigates the role of inflammation and other pathological processes in the progression of osteoarthritis and other musculoskeletal diseases. He and his research team are also exploring gene therapies as a strategy to reverse disease progression and prevent associated pain and motor symptoms. Prior to establishing his laboratory in Canada, Dr. Grahl worked at Baylor College of Medicine studying gene therapy. Our second speaker is no stranger. Dr. Jose Luis Milan is a professor of human genetics of the program of Sanford Burnham Presbys in La Jolla, California. He's also a member of our SoftBones Scientific Advisory Board since its inception, and he spent 45 years studying the structure and function of alkaline phosphatase using the power of mouse genetics. He's developed the animal models that span the severity of HPP, and his infantile mouse model was instrumental in developing aspartase alpha. His current work focuses on validating the potential of gene therapy. Dr. Grahl, we'll start with you with a high-level overview of gene therapy, and we look forward to learning from you today.
Thank you for the introduction, Deb, and good evening, everybody. I'm just going to stop here for a second. All right, good evening, everybody. Thank you for the invite from SoftBones to kind of go over an overview of what gene therapy is. And Dr. Milan will follow me in terms of talking about how it might be applicable to HPP. As Deb mentioned, my work lies predominantly in the treatment of arthritis, specifically osteoarthritis, and the use of gene therapy for that disease. But as you'll see throughout my section of tonight's webinar, gene therapy is applicable to many different types of diseases, one of them being HPP. So if we're going to talk about gene therapy, we have to first talk about what is a gene. So as just an overview, this is an image of, or sort of an animation of what one of our cells would look like. And our cell is composed of essentially two sections. The nucleus that houses the instructions, or the DNA, that codes for all of the proteins that make up all of the components of our body. So the keratin in our hair, the enzymes in our stomach that help us process our food, all of the blueprints for that are encoded in our nucleus. And I'm just going to activate my pointer here. In our nucleus, in our DNA. And then the other section of our cell is the cytoplasm, and this is sort of the factory section. So this is the section of our cell that takes those instructions and turns them into protein and secretes them into our body or into the various tissues of our body. Now, the DNA in our cells is quite large, so it's compacted into these structures called chromosomes. So within our chromosomes, we have our DNA, and within our DNA, we have these sequences that we call genes. And the genes in our DNA are what code for those proteins like keratin and the enzymes in our stomach. And these genes are essentially the basic units of heredity. So it's the combination of the genes from your mother and your father that dictate the color of your eyes, that dictate the color of your hair, that dictate your height, as well as a number of other things. So the concept went, back decades and decades ago, when the structure of DNA was discovered and genes were discovered, it sort of initiated this idea that when we're thinking about diseases that are caused by mutations in these genes, that maybe we could use these genes themselves as some type of therapy. So what does a gene look like? For the purposes of our discussion tonight, a gene is composed really of three major sections. So these dark blue sections we call exons. This is the portion of a gene, the DNA, that encodes for that final protein. And the expression of these exons, or the transcription of them into the message or the RNA, which is required to make that protein, is driven by this on-off switch that we refer to as a promoter. So to turn any gene on in your body, you need to activate it through this promoter structure in our DNA, which will then take this sort of instruction, the instructions encoded in our exons, turn it into a message that we call RNA. And it's this message that's read by our cells to generate the different proteins in our body. Our DNA is also composed, or these genes are also composed of these red sections that are called introns that actually don't encode any sort of instructions for the protein, but are mainly involved in regulating the gene along with the promoter. So the two things that you really want to keep in mind as we talk about turning a gene into a therapy are that we need this sort of on-off switch to turn the expression of that gene on, and we need the instructions, which are encoded by these exons. So what is a mutation? So a normal gene in our body, let's say the gene that encodes keratin, which is the major protein in our hair, essentially is the instructions to produce the normal protein. So the normal gene for keratin produces the normal protein for keratin. A mutation essentially can have two effects. It can either create a protein that doesn't function properly, or it can lead to the complete loss of that protein. So those are sort of the two major outcomes. So a mutation in this sequence of instructions encoded in our gene, a change even in one of those messages can lead to the formation of this abnormal protein or a complete loss of protein. And so the idea of gene therapy is that we know that the normal gene works. And we know that in many of these monogenic diseases like HPP, we have a mutated gene. And the concept was, could we take the normal gene and replace the mutated gene with the normal gene and essentially cure the disease? And so gene therapy refers to the strategy where we take genes and we deliver them using a carrier that we call a vector in order to either treat or cure whatever disease we're talking about. And there are three strategies that we can think of when we think about gene therapy. We can think about replacing the mutated gene with the normal gene. And this would solve either the production of abnormal protein, or it would restore the production of that protein if it was lost. Alternatively, we can either add a gene or silence a gene to do the same thing and correct the disease. So in cases of diseases like Huntington's disease, where the abnormal protein actually leads to deleterious effects in our body because it accumulates, we might want to silence the production of that abnormal protein. Or in the case of some other diseases like HPP, where we have a loss of protein, we can add a copy of the normal protein rather than replace the mutated protein, and do the same thing, in a sense, rescue the disease and cure or treat it. Now, an important difference, important point to talk about is what are we targeting when we do gene therapy? And we essentially have two major types of cells in our body. We have what we call somatic cells, and these cells make up all of our different tissues in our body except for our reproductive cells. These are neurons, these are bone cells, these are the cells of our pancreas. These are cells that contribute to specific functions and specific organs, but that aren't responsible for reproduction. So if we treat disease by using a gene therapy in these cells, we correct that disease in the person we treat. But because we're not changing the gene in one of our reproductive cell types, like the sperm or the egg, that change is only effective in the person we treat. It's not passed on to their offspring or their children. So that's an important point to keep in mind as we go through the talk, is most of the time when we talk about gene therapy, we're talking about this somatic gene therapy. We're talking about treating the individual. We're not talking about correcting the mutation so that it doesn't affect their children or their offspring. And a major reason for that is because if we start, it's more bioethical in the sense that if we start manipulating the egg or the sperm or the embryonic cells that arise prior to the formation of the fetus, that technology could be used to manipulate a number of other traits. And so that there's sort of important bioethical concerns that we need to consider when we talk about germline gene therapy. So what we're talking about today is treating the tissues in the person with the disease and not their offspring. So that's somatic gene therapy. Now, the one way to do it is sort of the original idea, which is we want to replace that defective gene. Okay, so we have a gene that's defective. It's either producing an abnormal protein or it's not producing a protein at all. And we know the sequence and we know the structure of the normal gene. And so in this case, we do something called gene editing, where we cut the DNA around or inside that abnormal gene in our cells, and we put in the regular functioning gene. And so this is called gene editing. And in this case, we would essentially cure the disease because we would be fundamentally altering our own DNA and replacing that abnormal gene with the functioning normal gene, or the one that produces the normal protein. Now, currently gene editing therapy, there's no examples of a gene therapy that actually conducts gene editing in our bodies. This is something that's being heavily investigated right now with some emerging technology. But what we're going to focus on more for the rest of my section is gene addition or gene silencing. And this is particularly applicable to HPP. So gene addition is, we're sort of at the same starting point. We have a mutation in our DNA, this little red box here. So this mutation in our gene that leads to almost no production of the normal protein. And so this is what's causing our disease. And so gene addition therapy is a little bit easier because we don't need to cut our own DNA and try to alter it, but instead we can just add back the functional gene into our cells. It's almost, if you think about it, when you have a disease, most oftentimes you're given some type of drug, and you have to take that drug, maybe it's every day, maybe it's once a week, because your body doesn't produce it. In gene addition therapy, what we're doing is we're taking the normal gene and we're asking our cells now to be the drug producers. And we're asking our cells now to produce the normal gene to overcome the loss of function that we have because of our gene mutation. And so our own cells are now becoming the producers, the drug producers, and they're correcting the mutation. And again, we're not correcting the error in our DNA, but we're simply adding in the functional gene into those cells so that they can do their job more effectively. And underneath here are some examples of where this is now being applied in the gene therapy world for different diseases. So retinitis pigmentosa and spinal muscular dystrophy are examples of diseases where gene addition gene therapy has been successful. Ex vivo treatment of X-linked severe combined immunodeficiency disorder is another example of a disease that's benefited from a gene addition therapy. And then the other possibility is that the mutation in our genes actually produces an abnormal protein that's bad for us. So this abnormal protein maybe binds up other proteins, or it interferes with various cellular processes that are important for our normal cell function, and that's what's causing our disease. And in these cases, we can silence that gene that's producing that bad protein using genetic sequences that can essentially turn it off. And so this is called gene silencing therapy. And Huntington's disease is an example of a disease where this would be very effective, because in Huntington's disease, the abnormal protein is actually causing detrimental effects, not because of loss of function, but because of other things that it does to our cells. So what do we need for gene therapy? We need two things. We need our therapeutic gene. So in the case of HPP, this would be tissue nonspecific alkaline phosphatase. And we need something to get it into our cells, or what we would call the vector, or the delivery vessel. Now, when we're designing our gene therapy, we have some options that we can play with here. And scientists have spent decades for many different diseases trying to optimize these types of delivery systems. So we have our therapeutic gene. Let's say it's tissue nonspecific alkaline phosphatase. Remember what I said before, we need some type of promoter to turn the expression of that gene on. And we have some options here. We could design a sequence, an on-off switch that is always on. And that would be great for a disease where it's a loss of function, where that protein's not being produced and it needs to be there all the time. And so we can design a switch that expresses it all the time in the tissue that we deliver it to. In my research, where we're looking at diseases that involve inflammation, where we want to inhibit something only when there's inflammation present, we could do something that's called inducible gene therapy, where our gene is only turned on when we need it to be turned on. And then the final option that we have is a or tissue-specific gene therapy. And so in these cases, we use a promoter that's only present, let's say, in the liver, so that when we deliver our therapeutic gene to the liver, it's only turned on in the liver. And this is really, a control mechanism in case some of our gene therapy got out of the liver and got into another cell type that we didn't want it in. By using a tissue-specific or a cell-specific promoter, even if that happened, the therapeutic gene is only ever going to be on in the liver. So it's sort of a fail-safe mechanism in cases where we're worried that it might escape from the site that we injected it into. And then the final thing that we need is a delivery vessel. So our bodies are really good at identifying foreign DNA outside of our cells and breaking it down. So we can't just inject DNA into our bodies and expect it to get into our cells and produce what we need it to produce. So we need to protect it. So we need some type of carrier or vector or vessel. And there are two major options. There's sort of two major categories. There's viruses that we engineer to express our gene therapy. And then there are chemicals that we can use, like lipids and polymers, that we can coat our gene therapy in so that it can get into our cells. And we want it to get into our cells because that gene therapy or that DNA needs to go through the plasma membrane, which is the barrier outside of our cells, and get into our nucleus where it can be expressed, where it can turn on expression of that RNA molecule that will lead to the production of that protein that we want to produce. And so the vector also protects the DNA or the RNA or the nucleic acid molecule from our immune system, because our immune system, like I said, is very good at recognizing these molecules when they're outside of our cells. Now, once it's in our cell, we have to decide where we want it to be. And there's a couple, when we design a gene therapy, it's always pros and cons. So, and it's no different when we're trying to decide where we want that gene therapy to exist in our cells. So our gene therapy has to get into the nucleus. And one option is it can get into the nucleus and it can live outside of our normal DNA. And that's great for gene addition therapy, because it's still producing the gene that we want, the protein that we want to produce, and it's not disrupting our own DNA molecules. The problem is that all of our tissues turn over, all of our tissues repair themselves, all of our tissues replace old cells with new cells throughout our entire life. And when the therapeutic gene lives outside of our own DNA, when our DNA replicates itself to make daughter cells, that gene therapy doesn't get regulated. And so what can happen is essentially it gets diluted over time and eventually lost. And so what this means is that, we might deliver a gene therapy and it may last for a long time, maybe a year, maybe 2 years, but eventually it might need to be re-administered, okay? Now, the other option is we can design our gene therapy to insert directly into our own DNA. So we use something that can insert our gene right into our own DNA, into a sequence of our DNA that doesn't disrupt any of our other genes. And in this way, every time our DNA replicates, so does our therapeutic gene. So even though our tissue is turning over, our therapeutic gene is always there producing the protein that we want it to produce. So this means that after one injection, we can get really stable expression of the protein that we're trying to use to treat our disease. So in this case, it might be tissue nonspecific alkaline phosphatase. The problem, though, is anytime we insert something into our DNA, we might disrupt some of our other genes and we might cause disease because of those disruptions. Scientists have gotten really good at making sure these insertions don't happen close to any of our other coding genes in our DNA, but still there's always that possibility. So when scientists are designing a gene therapy, it's again, these pros and cons of are we okay with injecting maybe once every 5 or 10 years? Or do we want something that lasts, but we take the risk of maybe disrupting our own genetic material? And those are things that we have to consider. Now, non-viral delivery systems, again, remember we have to coat our DNA to get it into our cells, and we can do that using non-viral systems like lipids or polymers are really good because they're super easy to produce. And they're relatively low, they show us relatively low toxicity. So they're not overly damaging to our bodies at low doses. The problem with these, though, is that they're not very efficient. So they can get the DNA into our cells, but they don't do a fantastic job. And they really only work at the site that we deliver them to. So they're not very good at deciding going only to the liver or only to the bone or only to the muscle. They sort of just go wherever we inject it. So they're not overly specific. Now, a solution to that is to take advantage of viruses, which have evolved over hundreds of thousands of years to become really, really good at infecting our own cells. And I know that we're in the middle of a global pandemic, so this may be a little bit sensitive, but I'll do my best to talk about it as a tool. So taking these viruses and using them, using their coatings, which are designed to do a really good job of binding to our own cells to deliver our gene therapy. So they're really good because they're really capable of delivering that gene therapy product to our cells. They're really good at making sure that it's expressed really highly and it's expressed in the cells that we want to infect. So some viruses, if let's say we wanted to infect our lung, some viruses are better at infecting our lung and other viruses are better at infecting our muscle. And by using different types of viruses, we're able to make sure that our gene therapy product only goes to certain types of cells. The major disadvantage with viruses is that a lot of viruses we've seen before. So these viruses that we use oftentimes are some of the same viruses that we've gotten a cold from, or we've gotten a stomach bug from, and so our immune systems have seen them. And so a disadvantage of some of using viruses can be that our immune system might actually recognize the carrier and eliminate it before it can actually get to our cells. And so there are ways to deal with this. We can sort of suppress our immune systems with an immunosuppressant until we're sure that the virus is in our cells. But that's sort of one of the major disadvantages of viruses. Now, when you look at gene therapy, there's lots of options. So I know it might feel overwhelming because it's just options, 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 but there's different reasons we might use different types of viruses. Adeno-associated viruses are really small viruses. Their disadvantage is that they can't hold a lot. So if we've got a big gene, adeno-associated viruses just might not work. But the major advantage here is that there's no known human disease that's caused by adeno-associated viruses. So we know that if we give an adeno-associated virus, we're not going to cause any type of disease. Now, adenoviruses are a different type of virus. They can handle a lot more material. So in my lab, we can actually put several genes into one adenovirus and do combinatorial therapies. So there's a lot of options. We're able to deliver multiple drugs in one virus, and for osteoarthritis, that's a great thing. The problem with adenovirus, though, is that it is associated with human disease. And so it's something that we have to keep in mind. We really need to make sure that this virus is not able to cause disease when we deliver it with our gene therapy or our therapeutic gene. And finally, we have this class of virus called retrovirus or lentivirus. And this virus is unique because these two viruses, when they deliver the gene therapy, that gene therapy sits outside of our DNA. It never enters our DNA. So it's that gene therapy that if our tissues divide, that gene therapy will become less and less and less until it's lost. Retroviruses are able to insert our therapeutic genes directly into our own DNA. That's how they replicate their own genomes when they're a virus that causes disease. Like I mentioned before, that's fantastic if we want to ensure that the expression of our therapeutic gene is there forever. But the possible disadvantage is that if it inserts near one of our other regular genes, we might cause disease, oftentimes cancer. So it's something that we need to carefully consider when we are trying to decide how we want to deliver our gene therapy to ourselves. Now, when I say we use a virus, we really are using the outside of the virus. So we're using the shell of the virus to trick our body to get our gene therapy or therapeutic gene inside of our cells. And so what we do when we're designing a gene therapy is we actually take the virus's own genes and we remove them. So we take all of them out, and in place of those, we put our therapeutic gene. And we do the same thing in the adeno-associated virus as we do in the adenovirus. We take those viral genes out, and we only put in our therapeutic gene. And what that means is that any of the genes from the virus that might cause disease are now gone, and only our therapeutic gene is present. So we've been able to avoid causing, a cold or a stomach problem using the adenovirus by essentially removing all of its own genes and only putting in our therapeutic gene that we're interested in introducing to our body. And so we've designed our gene therapy now, we want to get it into our cells. So how do we do that? And so there's two types of gene therapy that you'll hear of. There's in vivo gene therapy and there's ex vivo gene therapy. And in vivo gene therapy really just means that we're going to take our gene therapy, maybe it's in a virus, maybe it's covered by lipids, and we're going to inject it directly into our tissue, okay?
This is one arm, right?
Ex vivo gene therapy, though, means that we take our own cells out, we modify our own cells outside of the body in a dish. So maybe we correct that mutated gene in our cells outside of the body in a dish, and then we put those cells that have now been corrected back into our tissue or into the patient. So in this case, we're modifying the cells outside of the body and we're putting them back. And in vivo gene therapy, we're delivering that gene therapy product directly into our cells in the tissue. So we're not taking any cells out. And we can introduce it either directly using physical methods like injection into the muscle or the liver, or we can use physiological routes. We can introduce it into our bloodstream and allow it to be carried throughout our body, or we can introduce it into our respiratory system if it's a mutation that exists within our lungs or our airways that we want to correct. The important thing is that, a couple decades ago, gene therapy was sort of this idea that emerged after this discovery of the structure of DNA and the fact that genes contributed to both disease as well as to hereditary traits. But in the last 20 to 30 years, gene therapy has really exploded. in terms of both gene therapy products that are in clinical trials, as well as approved gene therapies. Now, if you look at this diagram, you'll see that the large majority of gene therapies have been developed for cancer. But what we're seeing now is that almost 10% of these gene therapies have now been developed for monogenic diseases. So diseases caused by a single mutation, just like HPP. And I'm just going to leave here with a list of some of these gene therapies that have been approved either in Europe or in the US, to give you a sense that we're not so much at the beginning of this new frontier of medicine, but we're sort of at that exponential growth phase. So there have been gene therapies that have been approved, they've been administered to patients, they've been successful. And so what we're going to see in the next 10 or 20 years is more and more gene therapies being developed to address some of these diseases like HPP, to allow us to treat the disease, to subject patients to fewer injections and things like that, to improve overall quality of life. And so with that, I'm going to let Dr. Milan talk a little bit more about the direct implications for HPP.
Okay, can you see my slides? And I am unmuted now. Is this okay?
Yes, we can see your slide.
Wonderful. Very good. So thank you very much, Dr. Grohl. That was a wonderful introduction on gene therapy. My task today will be to review what has been published to date on gene therapy efforts for hypophosphatasia. And Dr. Grohl already described the two methods that have been used to date, and that is ex vivo gene therapy and in vivo gene therapy. Ex vivo, to recapitulate, are cells isolated either from peripheral blood, bone marrow, that are expressing a functional copy of the missing gene or defective gene. These cells are expanded in vitro and then reintroduced into the patient. And this can be autologous, meaning from the same patient, the same subject, or allogeneic, meaning they come from an unrelated or not immune identical patient or subject. Examples of these cells are mesenchymal stem cells. These are cells that essentially can produce bone cells, cartilage, tendons, muscle, and so forth. Hematopoietic stem cells that can recapitulate the variety of cells that we have floating in our blood, leukocytes, erythrocytes, platelets, and so forth, and immune cells. In vivo, a functional copy of the gene or cDNA is packaged into a viral vector for delivery to the patient, either systemically, injecting it in the blood so that it distributes throughout the body, or locally into individual tissues. And examples of these are injections into skeletal muscle, into liver, heart, eye, brain, etc. So I will review to you what has been done to date. But let's get back to some of the basics of HPP. When we talk about the skeletal defects of HPP, what are those? Well, the missing enzyme, or in this case, the malfunctioning alkaline phosphatase, is not able to hydrolyze inorganic pyrophosphate, a very potent inhibitor of calcification. So that, I hope you see my cursor where I'm pointing, Debbie. The extracellular matrix, this collagenous fibers here, should be all black with bone mineral, with hydroxyapatite. And because the enzyme is not destroying pyrophosphate, you have this accumulation of the inhibitor and then suppression of mineral deposition in the extracellular matrix. So the enzyme here is missing or is malfunctioning at the surface of osteoblasts, bone-forming cells. It's also malfunctioning at the level of matrix vesicles, these blobs, which is where mineral gets initiated. And that's what allows pyrophosphate to accumulate and suppress mineralization. So cell therapy aims at bringing in normal cells now that will have a functional alkaline phosphatase to be able to hydrolyze pyrophosphate and allow mineralization to proceed. Now, the first trials were actually done by Dr. Michael White on the animal model that we had developed in the mid-90s. He brought them to his laboratory and did some proof of concept studies in the mouse model of infantile HPP, and then moved on to some patients that he was treating at the time. So the initial results were like this. He took bone marrow cells from unaffected mice, wild-type mice, and injected them into the knockout animal, the model of infantile hypophosphatemia. And then he saw that leukocytes were detected in blood smears of the treated mice, indicating engraftment. And this was very promising. All mice survived to 44 days, which was the end of the experiment, with skeletal improvement. So this early mouse work was very encouraging. And then he moved on to treating an 8-month-old girl with infantile hypophosphatemia, went into cytoreduction to eliminate endogenous cells to make room for the injected cells, was given T-cell depleted identical marrow from her sister, and achieved engraftment, but needed marrow-enriched mesenchymal stem cells repeated treatment after 6 months. But there was prolonged improvement of skeletal condition, and she survived, but remained small. The other paper that he then published in 2007 was another child. The child, once again, immunotolerated, and there was a combination of bone marrow cells, engrafted bone fragments from the father, administered subcutaneously or intraperitoneally, and then osteoblasts grown out of those bone fragments. And then 4 months later, the radiographs indicated that there was improved skeletal mineralization, and he reported that the patient, now 8 years old, 7 years after transplantation, was active, growing, and had clinical phenotype of a milder form of hypophosphatemia instead of the severe form of hypophosphatemia. So a rather successful first trials for cell therapy for this disease. Another paper, an abstract and a paper, were published by Dr. Taketani in Japan. And here again, there was 2 patients that were treated with allogeneic mesenchymal stem cells. The donors were asymptomatic relatives harboring heterozygous mutations for the APP gene, so they were carriers. The patients underwent bone marrow transplantation after myoablation, again, conditioning of the patient, and then that followed mesenchymal stem cell transplantation of cells expanded ex vivo. The patient survived, restoration of alkaline phosphatase was achieved, but normal bony architecture could not be achieved. So there were still deficits in the skeleton. So now if we moved on to gene therapy itself, what is the principle here? The principle is to try to deliver the missing enzyme now to the bone matrix, not to the cells, but to the bone matrix where it is needed in order to try to destroy inorganic pyrophosphate to allow calcification to proceed. Now, this is the principle of aspartate alpha that many of you may be under treatment. The problem is that this is a very large molecule, 726 amino acid, and it is just too large to be encoded by a viral vector. So it had to be trimmed down to size. The way to do that was to remove the FC region of immunoglobulin, which was artifactually put in to increase half-life of the biologic in vivo, and also to allow for rapid purification in the laboratory. But this moiety is not really necessary when you're asking the body to produce the biologic in vivo. So FC was removed, only the polyaspartic acid motif, which is really the targeting motif that allows the enzyme to be anchored to hydroxyapatite, was left. And this is the molecule that was essentially inserted into viral vectors for the viral vectors to encode it and translate it in vivo. And what has been done? Most of this work has been done in the laboratories of Professor Shimada and Miyake, both gene therapy experts in Japan. This is a collaboration that we started more than 10 years ago. In fact, Professor Shimada was present at the conference in Unang, France, where the preclinical work on aspartate alpha was presented. And then we discussed the possibility of beginning to try gene therapy in the infantile animal model, the mouse model. The first paper that was produced was in 2011, 2 papers in 2011, and going down now to last year, where 2 papers were also presented. And let me show you some of the summary of what this work has revealed. In the very first paper, we used a lentiviral vector. Remember the lentiviral vector from what Dr. Grohl said, integrates. So it's a very good research tool. It's not an optimal vector for in vivo delivery to patients, but it's a very good research tool. So in this first paper, this lentiviral vector containing the TNAP-D10, the mineral targeted form of alkaline phosphatase, was introduced via the jugular vein, so systemic administration, into 1 to 3 day old knockout animals. The animals all survived to up to 160 days where the experiments were interrupted. Circulating levels of alkaline phosphatase went up 73-fold compared to those in untreated and actually in reference wild-type animals. The vector distribution is interesting here because this is systemic delivery of the virus. The virus mostly went to the liver, then also to the spleen, equally to heart, to kidney, to lung, to bone. Essentially, the vector goes to most of the tissues in the body and turns these tissues in the body as factories of mineral targeting alkaline phosphatase. In these trials, there was improvement in skeletal mineralization. So everything went well, but of course there is the risk of insertional mutagenesis that Dr. Grohl already described. So the idea was try to get away from lentiviral vectors and move into what was considered safer vectors. Well, so then the next paper, also in that year, was the AAV, adeno-associated viral vector 8 in this case, which is episomal. It stays in the nucleus outside, does not integrate in the genome. The vector was injected also in the jugular vein, so again, was systemic injection. The twist in this paper was that we expressed 3 different forms of the enzyme. The mineral targeted alkaline phosphatase, very comparable to aspartase alpha. Alkaline phosphatase with an epitope tag, with a small tag that also confers some binding to mineral, about 20% of that is conferred by D10. But interestingly, also non-modified alkaline phosphatase, meaning the alkaline phosphatase that normally would be expressed on the cell surface and on the surface of matrix vesicles. And even this therapy worked quite well. 40% of the animals injected with a non-modified enzyme survived. This opens up possibilities for the future when we will try to target other tissues besides the skeleton for improvement of hypophosphatasia-related deficits. The vector was also distributed throughout the body to heart, muscle, lung, liver, kidney, spleen, bone, again, because it was systemic administration. The next paper was quite interesting as well. It was an attempt to do in utero gene therapy. And for this, the mice, there was a midline laparotomy in the mice at 15 days of gestation. And then each fetus was injected. They underwent transuterine intraperitoneal injection of the viral vector. And the vector was detected in most tissues, but was mostly concentrated in the brain and bone compared to postnatal injections of the same vector. We again saw enhanced mineralization and based on X-ray images of chest and paw. The activities were high, but not that high. Remember 73-fold in the other trials. Here was about 10-fold increase in alkaline phosphatase activity. So then the next step in the development was try to limit expression of the vector to only one tissue, in this case, to muscle. One way to do that was to use a muscle-specific promoter to drive expression of the therapeutic gene, using this creatine kinase, muscle-specific creatine kinase promoter. The skeletal muscle is an important candidate as reservoir for vector-mediated enzyme replacement because of the large mass, is easily accessible, and it has high vascularity. And the other attempt that was made was, again, because of bilateral injection of the quadriceps, you would expect the viral vector to stay in the muscle and not disseminate throughout the body to try to reduce inadvertent germline transfer that the systemic injections could lead to. The vector, again, was detected primarily in muscle, quadriceps, where the injections were done, also in heart, liver, bone, and bone marrow. So much more limited distribution of the vector. There was 15 microliters that we injected, and that was still too high, and that may explain this enhanced distribution. TNAP activity was increased only 10-fold, and that may explain why even though there was increased survival, there was still insufficient mineralization and still abnormal bone structure in the mice. So the last two papers from last year were actually an additional improvement. Here is a single injection of this vector in the quadriceps femoris. Injection was restricted to 2 microliters, so very small volume to try to keep the vector right there at the injection site. The activity reached was much higher, now 100-fold higher activity up to 18 months. Now, with extremely high doses of vector, we could observe ectopic calcification in the paws in some of the soft tissues, but ectopic calcification was not observed if you kept the vector dose down to about 3 × 10^11 vector genomes per animal. So this is something that one can manipulate, the amount of vector that is injected in vivo. The mice had normal physical activity, healthy appearance, and in fact, the treated animals were undistinguishable from wild type unaffected animals. So this is all very encouraging. And finally, also from Professor Shimada's group, there was an attempt to do a combination of cell therapy and enzyme replacement therapy. In these two posters, and then in this paper, so lentivirally transduced bone marrow cells expressing the mineral targeted TNAP were introduced back into the animals. And the treated mice had normal appearance and had no seizures. The transduced cells engrafted efficiently and supplied up to 400-fold levels of activity. And it was continuous for at least 3 months of the observation window. The overexpression of mineral target D10 did not affect the differentiation of bone marrow cells into the multi-lineage that needs to be reconstituted. And plasma activity was sustained, and activity was found also at the bone surfaces. Remember, this is a combination of cell therapy where some cells are going to go to the bone and begin to act in the bone. And then you have the enzyme replacement. These bone cells are also going to be secreting bone mineral targeted alkaline phosphatase that is going to go to the matrix to further sink pyrophosphate concentrations. And no ectopic calcification was observed in these trials. So I'm going to finish by addressing head-on what Debbie Fowler asked us to address, what it is and what it is not, what is gene therapy and what it is not. So what are the pros of cell therapy using either mesenchymal stem cells or hematopoietic stem cells? The pros are intrabution infusions. It could be one or few. There is plenty of clinical experience in other diseases with bone marrow cell transplantation. There is no risk of germline gene transfer. There is no immunoreaction against the viral vector. These are all good things. What are the cons? Well, the degree of engraftment is limited. So repeat injections may be required. Conditioning of the patients is required. So what is cell therapy? It's a therapy to replace, complement endogenous defective cells by new functional cells in mineralizing tissues. That is what has been done today in HPP. If the mineral targeted alkaline phosphate is overexpressed, there is the added element of enzyme replacement in addition to the cell replacement. What it is not an attempt to replace a defective gene by a functional gene in all cells of the body, as other alkaline phosphatase deficient cell types are not replaced nor replenished. What is the viral vector delivery of mineral targeted alkaline phosphatase for HPP like? Well, the pros are, in mice, a single injection for life. This may not be so for humans, but certainly for extended periods of life, rather than the multiple injections per week that patients under aspartate alpha treatment must undergo. What are the cons? Well, it is difficult to control and restrict the distribution of the viral vector to one tissue, as we saw. There is potentially expression of the viral vector may be reduced with time, so that follow-up injections may be needed. There is a potential for anti-serotype antibodies that may prevent therapy in subpatients. There may be vector shedding into biological fluids. And it's hard to control overexpression if that were to be the case, if that were to be a problem. So what is viral vector delivery? is an enzyme replacement. So what has been done today with TNAPD10 is enzyme replacement, but where the viral vector delivers the instruction to the patient's own cells to produce mineral target TNAP. And what it is not an attempt to replace a defective gene by a functional gene in all affected cells. Now, why do I say this? Because clearly, to date, we have been focused on the skeletal dental phenotype or manifestations of hypophosphatasia, but alkaline phosphatase is present in other tissues where it also performs important functions. Now, we are just beginning to understand what the long-term consequences of a deficiency in the enzyme in other tissues might be, but we're not yet attempting to replenish the enzyme in those other tissues, the immune system, kidneys, liver, brain, even fat cells. So this is still, there is a lot of room for investigation in this arena. And with that, I think I will stop and invite your questions.
Thank you. That was so incredibly in-depth and thorough, and we're so grateful for that overview and for the specific potential applications in HPP. We do have a couple questions that came in. One of them is using bank stem cord blood or tissue an option in gene therapy? For example, if somebody banked cord blood for maybe a child who does not have HPP, is there any application for that cord blood?
I'm not a physician, but I would estimate that yes, indeed, that would be a wonderful source of mesenchymal stem cells, or even just whole blood collection of cells. But perhaps Dr. Grohl may have a better answer than me.
Dr. Milan said what I probably would have said. I think it's a good source of stem cells, certainly talking about the ex vivo gene therapy approaches where the functional gene is introduced into the cells and then the cells into the patient. It certainly would be a source of cells for that application. Yeah.
Great. Another question that came in. If we turn on a gene that produces a certain level of ALP, will it still modulate?
Right, that is a very difficult question. In fact, the best one can do right now is to insert the cDNA. The cDNA, almost by definition, doesn't have regulatory elements. So it's just a message for the cells to produce TNAP. We don't have any inherent way of modulating it up or down. And this is why in one of my slides I mentioned this. We do not know how to regulate it in case that there is a dramatic overexpression. Ideally, one would develop a system with an on-off switch, as Dr. Grohl already mentioned, but that remains to be conceived and conceptualized and developed.
I can add a bit to that.
Sure.
It's you know, it's part of what contributes to decades of trying to fine tune these therapies before they go to patients. But you know, not every gene in our body is expressed at the same level. Some genes are expressed at very low levels, other at very high levels. So can you modulate? We don't have a really good way of allowing the gene to modulate itself, to detect its own levels and shut itself off if it's too high. But we can try to control dose by picking promoters or those on-off switches that may turn on the gene only a little bit, or a mid amount, or a really high level. So the promoter sequence is very important in allowing us to try to fine tune where, when, and how much the gene is turned on. But to know how much we need to turn it on to correct or reverse the disease in the patient is another big question too.
Makes sense. Okay, another question that we had, any indications potentially for gene therapy in utero?
Well, at least in the mouse model, it is possible. However, probably we are far away from clinical trials in utero. We are just beginning to conceptualize clinical trials in adult patients. So that is it for the future.
I think we'll struggle with the coming back to the bioethical ramifications of manipulating our germline cells to try to stop a disease passing off onto our our offspring or our children, is that, you know, approving that type of therapy opens sort of a floodgate for things that we may not want to allow to be changed, right? So I think that manipulating our germline or manipulating in utero is probably a little bit of a ways away just because of those concerns. And gene therapy is certainly more on the forefront for treating the individual patient once they have the disease.
Okay, thank you. Another question. Do you think subsequent injections will have the same lasting effect as the first dose, or the immune system may interfere and reduce the efficacy?
That's a very good question. The short answer is we do not know. However, from asfotase alpha, we know that there is some immune reaction against the biologic, but that still has not caused major problems or disturbances in the treatment. So it is to be expected that a second booster may work just as well as the first, but the short answer, we do not know.
Okay. I think the thing to consider there too is our immune systems might recognize like a viral vector the second time we try to go and deliver it, for sure. But if even 20% of them get through to our cells, or 30 or 40, if they're producing a lot of the protein that we need, it may not be an issue. And certainly injecting into a tissue like muscle is probably much safer in terms of an immune reaction than in delivering it to our circulatory system, where there have been, you know, sort of more there have been sort of more extreme reactions in the past in patients. So I think, you know, the current experiments looking at delivering the viral vectors to muscle are probably a much safer alternative than the intravenous. And we can always change the virus type too. If we're talking about viral vectors, we can always change the virus type to something that we've never seen before. Or like Dr. Milan was talking about, you could do a cell therapy where your body's not seeing the virus at all. It'll just depend.
Great. Another question, can you explain what conditioning the patient means? It was one of the disadvantages noted by you, Dr. Milan, at the end of your talk. So just wanted to get a better understanding of what is meant by that.
Well, not being a physician, I've only worked with mice, and in mice, what we do is irradiate the mice, essentially to make room in the bone marrow for the injected cells to colonize and replenish that population of cells, be that hematopoietic stem cells or be that mesenchymal stem cells that are going to turn into bone cells. But essentially, you ablate the marrow with careful dosing of radiation to allow that to proceed. In case of patients, I understand that they use a certain regime, chemotherapeutic regime, to do the same. And the regimes are getting easier and easier to tolerate, but you still need to condition the patient, meaning to make room for the administered cells.
One final question. I think this is one that every patient has in the HPP community, and that's, you know, how far are we away from the trials? How far away are we from being able to access these kinds of therapies? The million-dollar question. What is your advice to patients who are, you know, kind of waiting on the edge of their seats for these trials to begin?
Well, right now you have a life-saving treatment. I will not shy away from that, because that has been a dramatic change in how HPP is being viewed. Gene therapy is still in the horizon. It may be 2 years away, 3 years away. There are companies getting interested in gene therapy, so things are happening, but it's still in the future. It's not next week we will not have a treatment, but perhaps in a year or two.
Right. Well, I want to thank you all again. I really can't think of a better way to spend Rare Disease Day than talking about future potential treatments for HPP and providing hope to all the patients who are listening. And we're just so grateful for you sharing your expertise. So thank you so much.
Thank you for inviting us.
Thanks for having us.
Yeah.
Thank you everyone for attending.
And a special thank you again to Ari Vand and to all the patients in our community who've made this presentation possible. Have a great night, everybody.